Hi, my name is Jared Hedrick. I will be presenting my enzyme lab poster, and I did that with Stephen Kirshner and Arman Mashasa. So we're going to be talking about catalysts today, more specifically biologic catalysts, which are called enzymes. And an enzyme is going to be, ours is our hydrolysis, and what that does is it uses water to tear apart two chemical bonds. And in this instance, we are going to be carrying apart peptide bonds from proteins. So how an enzyme works is it lowers the activation energy of a specific chemical reaction so that the less energy it needs, the faster it can produce whatever it's trying to produce. And it can help things happen a lot quicker, especially in our bodies when it needs it. So the one we're going to be talking about today is trypsin. And what trypsin does is it takes two peptide bonds and it breaks it down. Or it takes protein down and breaks it into peptide bonds. And it does this at a faster rate than our body normally would, which is why it's good for us and other organisms to have. So what we were presenting is if salinity affects how trypsin reacts with our body. So we increased our salinity levels, and we were going to see if the higher increase would be if it produced less or produced more, or opposite, if less salinity produced more or less. So the question we proposed was, does salinity affect our body? And our hypothesis was that the higher the salinity goes, the less effective it is going to be at being a catalyst. And so for our actual experiment, what we had was 7 centrifuge tubes, and we filled those with different buffers of salinity. So this ranged from 0 to 4.8. We're expecting the 0 to produce the most, and then 4.8 to produce the less, and everything in between. The exact salinities for the 7 centrifuges we used were 0, 0.3, 0.6, 0.9, 1.2, 2.4, and 4.8. And then I'll explain in a second which ones of those we used for our negative control group. So with that, we had to add 500 microliters of the azocasein, and the azocasein in this part is what is the protein that we are breaking down with trypsin. After the azocasein, we put the trypsin in. We put the trypsin in last, or we put the trypsin in before incubating, so that as soon as we put it in and we start putting it in the environment, it needs to be at the exact temperature, which was 38.6. That it is breaking down immediately, and we're not waiting around and losing time and all that. So we chose 38.6 degrees Celsius for our temperature and our environment, and that was due to a past experiment that another group had done where their range was roughly in the spot for the most effective environment for the trypsin produced as far as temperature was concerned. So that's why we used that one. We also added, after the incubation of 20 minutes and after we gave time for the trypsin to break down the azocasein, we added 10 microliters of TCA. And what TCA does is it actually stops the reaction between the trypsin and the azocasein. And this is a good thing, so we have that 20-minute window where it can actually break down in the environment we want it to, and then we can stop it. We don't have to, have it go for any longer, and then while we're using the substrate, actually have it continue to do the reaction. So this stops it, so we have an exact window and exact time, which is good. So after we did that, we centrifuged it on max speed for 2 minutes, and what the centrifuge does is it brings down the pellet that is actually inside that we couldn't get out through normal filtering. It subtracts the substrate at the bottom and it has the supernatant at the top. So basically what we're trying to do here is we're trying to get the supernatant alone, so we can take the absorbance of that, because that's what we're worried about. We're not worried about all of the extra products that the trypsin and the azocasein create inside of our centrifuge. So in that, we did that, and we did 4 trials of that, and we found it useful to do 4 trials because with 2, if there's a varying result, you're not sure which one it is. And for 3, you can have 3 varying results, but also getting an average of one of those if you have to get rid of is not possible. So we did 4 just to be safe, and we'll get to why we can do more later. For our negative controls, we did 3 centrifuge tubes, and we did 3 trials of that. We found it useful to do 3 trials. Again, we were pretty sure of the negative control because we know the actual effects of trypsin was going to be it, but just to make sure we had that in there, we did 3, and we did it at 3 different salinities as well. We did it at 0, 0.6, and 4.8. So kind of the radicals of each end, and then a middle spot that we thought was a comfortable position for it to actually, if it was going to produce any trypsin, do that. So that is why we did that, and it was the same process, just only doing 3, and instead we did not add any trypsin at the to react to. So we can see in our results later that made a big difference. And again with the centrifuge, and then we centrifuged it, even though the supernatant was very small because it was predicted. We still had to get as much as we could out before we put it in the spectrophotometer. For our results, we can see our hypothesis was roughly correct. So for 0, we had the highest absorbance at 0.734, and at 4.8, our highest salinity, we had our lowest at 0.36425. In the middle, there is some ranges. It starts to go up a little bit, and then you can see it drop and quickly rise again, and then go back down at a slow rate, and then it kind of plateaus once you get down to that 4.8 level. Our negative controls, we can see that barely any was produced. We have 0 being a 0.0063, we have a 0.066, and we have a 0.0374. So a very, very little amount was produced. And then we have our t-tests for all of these. The t-tests were very small. They go below our p-levels, so the difference in all of them, or sorry, they were very high, meaning the difference in all these trials was very different. We do have accurate results when it comes to that. And so our hypothesis was supported. However, we feel like we can take it a step further in saying that there will be a general trend, that we can narrowly specific cut down to what the actual what the ideal environment for the trypsin would be as far as salinity goes. So we see the peak at 0, and then it goes down, and then it comes back up. Roughly in between 0.9 and 1.2 is where it starts to drop off again. So we believe there might be another peak in between there. We had to put 0.6 and 1.2 to get to the 0.9. So maybe putting 0.6 and 0.9 together to get the number in the middle of that could produce an ideal environment. As well as doing more trials. The more trials, the better data you'll have, the more you'll have to work with, and that'll make our t-test more accurate as well. And then as far as knowing what other methods to do, we would have to get back to our classmates. Unfortunately, we can't right now, but if we wanted to do another round of this experiment, finding an exact pH would be great, and maybe using a buffer to put all those, because ours was at 8, and we couldn't really do much about that, unfortunately, because those are the ones that salinity came in. But the temperature, the more accurate theirs is, the more accurate ours can be in finding the most ideal outcome for it. Yes, and that's my poster presentation.